Unit 1. Viruses, General Account and Economic Importance. Structure. 1.1 Introduction Objectives. 1.2 Discovery. 1.3 General Account. Virus size. Structure of viruses. The virus symmetry. Viral envelope. The viral nucleic acid. Viral enzymes and other contents. Viral taxonomy. 1.4 DNA virus, T phage. 1.5 RNA virus, TMV, viruses, general account and economic importance. 1.6 Other virus-like things, subviral particles. Virions, prions. 1.7 Economic importance. Economic losses due to viral diseases. Economic benefits of viruses. 1.8 Summary. 1.9 Terminal questions. 1.10 Answers. Glossary. 1.1 Introduction. We all have suffered from symptoms of common cold that include runny nose, sneezing, feeling feverish, but not having fever, at some point of our life. The doctor does not prescribe an antibiotic, effective against bacterial infections, for the common cold, as common cold occurs due to an infection of virus, rhinovirus. Viruses are a unique class of infectious agents. They infect all types of organisms. According to the Carl Woese system of classification, the tree of life consists of 3 domains: archaea, bacteria, and eukaryota, figure 1.1. The archaea and bacteria are prokaryotic single-celled organisms whose cells have no definite nucleus. Cells of eukaryota possess a nucleus and membrane-bound organelles. The animals, plants, fungi, and protozoa are included in the eukaryota. You will be surprised to know that viruses are the only form of life which infects all the 3 domains of life, i.e. archaea, bacteria, and eukaryota. Viruses can be defined as small obligate, intracellular particles seen only by electron microscope that infect and take over the host cell to replicate. Figure 1.1, 3 domains of life. Viruses blur the boundary between inanimate, non-living, and life, living. Anything is considered living if it is made of cells, reproduces, grows, metabolizes, responds and adapts to the environment, e.g., bacteria, fungi, protist, human beings, plants, animals, etc. All viruses are acellular entities and are made of genetic material, DNA or RNA, inside a protein coat. Viruses do not have the ability to grow and multiply in any artificial media, as they are metabolically inert, inactive, and can be metabolically active only when inside a living host cell. They multiply or are metabolically active only inside an animal, plant, or microbial cell. This ability of the viruses to infect all forms of life makes them a major force for evolution, as each virus has the ability to introduce new DNA or RNA through shuffling and redistribution of genes into any other life form. Thus, it can be said that viruses have played a key role in shaping the history of life on Earth. In the first section of this unit you will learn how the viruses were discovered and the role tobacco growers of Europe played in this fascinating discovery. You will study how the ability of the viruses to defy the germ theory led to their discovery. You will also learn how inferences drawn from experiments by scientists led to certain concepts of virology. The structure of viruses and function of each component along with the properties of virus, will be studied in detail. Viruses are important due to the economic impact they have by causing diseases in humans, swine flu, animals, foot and mouth disease, and crops, tobacco mosaic disease, and the role they play in medicine, vaccine production, cancer therapy, phage therapy. In this unit, focus will also be on their economic importance. Objectives. After studying this unit, you will be able to describe how viruses were discovered. Describe and draw the structural components of viruses. Differentiate between helical, icosahedral, and binyl symmetries. Discuss the classification criteria of viruses and list examples of bacteriophages, animal and plant viruses. Draw and differentiate between DNA virus, T phage, and RNA virus, TMV, and enlist the economic importance of viruses. 1.2 Discovery. The credit for the discovery of virus goes to the invention of Chamberland-Pasteur filter, figure 1.2. Chamberland-Pasteur filter was developed in 1884 in Paris by Charles Chamberland, who worked with Louis Pasteur, father of modern microbiology. It consisted of unglazed porcelain candles, with pore sizes of 0.1 to 1 micron, 100, 1000 nm, which had the ability to completely remove/filter all bacteria or any other cells known at the time, from a liquid suspension. Through this simple invention a whole new science, virology, was established. Figure 1.2, the credit for the discovery of virus goes to the invention of Chamberland-Pasteur filter. At the end of the 19th century the germ theory of disease was formulated by Robert Koch, founder of modern bacteriology. The germ theory states that infectious diseases are caused by microorganisms. The causative agents of disease can be seen with the aid of a microscope, cultivated on a nutrient medium, and can be retained/filtered by bacterial filters. At about the same time, i.e., the end of the 19th century, tobacco growers in Europe were worried because the presence of mosaic-like pattern on tobacco leaves was causing poor crop yield and heavy losses. Some of the farmers even stopped growing tobacco, and a search for the causative agent responsible for mosaic-like pattern on tobacco leaves began. A German scientist, Adolf Meyer, 1883, did an experiment where he rubbed the sap extracted from diseased leaves onto healthy plants. The latter also developed the disease. Meyer was unable to isolate the causative microbe present in the sap. Therefore, he concluded that the causative agent may be very small bacteria which could not be seen by a microscope. Two scientists, Dmitry Ivanowski and Martinus Bajerinck, played a crucial role in the discovery of the causative agent of this puzzling disease, figure 1.3. Figure 1.3, Dmitry Ivanowski, 1892, and Martinus Bajerinck, 1898, are credited for the discovery of viruses. In 1892 Dmitry Ivanowski became the first person to show that the causative agent of the mosaic disease of tobacco plants, visible as a discoloration of the leaf, passed through a bacteria-proof filter, and could not be seen under light microscope. Dmitry Ivanowski performed an experiment to isolate the causative agent of tobacco mosaic disease, TMD. He crushed the leaves of the TMD-infected plants, and filtered the mixture through a bacteria-proof filter candle. To his surprise, the sap, which had passed through the bacteria-proof filter candle, was infective. From his observation, Ivanowski concluded that TMD was not caused by bacteria, but by something smaller than the bacteria that could pass through these bacteria-proof filters. Dmitry Ivanowski was, however, somewhat unconvinced by his discovery and doubted the efficacy of his bacterial filters. Another microbiologist, Martinus Bajerinck, repeated the same set of experiments in 1898 and observed that the causative agent of TMD passed through the bacterial filters and validated the discovery of Ivanowski. Moreover, he observed that the filtered agent could not be cultivated in non-living media as is expected for bacteria. He believed that TMD was caused by a new form of infectious agent which was smaller and simpler than bacteria. He termed it contagium vivum fluidum, or virus meaning poison. At the same time, Friedrich Loeffler and Paul Frosch, figure 1.4, were working on foot and mouth disease, FMD, a disease of cloven-hoofed animals, e.g. cow, buffalo, etc., which is characterized by vesicular lesions in and around the mouth, on the feet and on the teats, and causes high morbidity and reduces milk production. Loeffler and Frosch isolated the extract from the vesicles of infected animals and filtered it through bacterial filters. They diluted the filtered extract having the causative agent. On dilution of the filtered vesicle extract, the concentration of causative agent in the filtered vesicle extracts decreases. They then injected the diluted filtered vesicle extract in healthy animals. The healthy animals developed FMD, even after dilution. Loeffler and Frosch again isolated the extract from the vesicles of infected animals and filtered it through bacterial filters. The filtered extract having the causative agent was again diluted and injected in healthy animal. they observed that foot and mouth disease could be transmitted from animal to animal even after dilution. Loeffler and Frosch from their experiments concluded that the causative agent must be reproducing as it was infective even after dilution and so could not be a bacterial toxin but something living. Loeffler and Frosch were able to rule out that the causative agent of FMD was not a toxin and were able to discover the first animal virus. Figure 1.4, in 1898, Friedrich Loeffler and Paul Frosch showed for the first time that animal virus was responsible for foot and mouth disease in cattle. Bacteriophages, the virus infecting bacterial cells, were discovered independently by Frederick Twert in England in 1915, and by Félix de Herelle at Pasteur Institute in Paris in 1917, figure 1.5. Frederick Twert had been trying to grow viruses in the artificial media, which seems a futile path of research to us now, as we now know that viruses can only grow inside living cells and not on artificial media. Apparently, he never succeeded in growing the filter-passing agent on artificial media. He did, however, observe something else. He had inoculated an agar medium with some fluid, having vaccinia virus, commonly used for smallpox vaccinations. He noticed that although the vaccinia virus did not grow, however Micrococcus, a type of bacteria, did grow. The bacteria, however, appeared to be afflicted with some disease. Inoculated agar tubes often showed watery-looking areas, and in cultures that grew micrococci it was found that some of these colonies could not be subcultured, but if kept they became glassy and transparent. This phenomenon came to be known as the glassy transformation of Twert. Twert made some very interesting observations about the glassy transformation. The affected colonies would not grow on any medium. 2. Examination of the glassy areas revealed only minute granules and no bacteria. 3. If a pure culture of Micrococcus was touched with a small portion of one of the glassy colonies, the growth at the point touched started to become transparent and gradually made the whole colony transparent. 4. After filtration of the glassy material through a chamberland candle, it retained its ability to cause the glassy transformation. The transformation could be conveyed to fresh cultures for an indefinite number of generations. Frederick Twert published a report of a glassy transformation of bacterium Micrococci. Twert noticed that the Micrococcus colonies were being lysed by the ultrafilterable infectious agent. Twert referred to it as the bacteriolytic agent. This was the first evidence of a bacteria virus. In 1917 de Herelles rediscovered this phenomenon with his first communication on bacteriophage, an invisible microbe antagonistic to dysentery bacteria. The main observations reported were that an agent capable of killing the dysentery bacterium, Shigella, appeared in the intestines of persons recovering from dysentery and, that this agent could be filtered through a chamberland filter and still be active, that it could be serially transmitted and hence must be a living germ. Since the virus was incapable of multiplying except at the expense of living bacteria, he called his virus a bacteriophage, literally a bacterium eater, or phage for short. This is derived from the Greek word phagein, which means to eat. Thus the first definition of these new agents, the viruses, was presented entirely in negative terms: they could not be seen, could not be cultivated in the absence of cells, and most important of all, were not retained by bacteria-proof filters. Figure 1.5, Frederick Twert, 1915, independently discovered bacteriophage. In 1935 Wendell Stanley for the first time crystallized a virus, the tobacco mosaic virus, TMV, figure 1.6. The crystallized virus when injected inside host cell also showed infective properties, proving that viruses could exist as living entities in living cells and as non-living entities, crystals, outside living cells. Crystallization of virus opened up the debate whether viruses are living or non-living or are just on the threshold of life, see box. Figure 1.6, in 1935 Wendell Stanley, 1904 to 1971, a young chemist purified a virus in the form of needle-shaped crystals with the chemical properties of a protein. Box 1.1, are viruses living or non-living? The controversy whether viruses are living or non-living is continuing since TMV was first crystallized. Viruses are considered living as 1. They possess genetic material, DNA or RNA, and protein. 2. They can replicate only inside the host cell. They use the cellular machinery of the cell, ribosomes, tRNA, enzymes etc., to replicate its genome and synthesize protein code. 3. Even the crystallized virus conserve the living properties, and 4. They are transmitted from infected host to uninfected host. Viruses are considered non-living as 1. Viruses are unable to show living properties outside a cell. 2. They can be crystallized. 3. Viruses can replicate inside host but cannot grow, and 4. They do not have metabolic machinery. Box 1.2, Invention of Vaccine by Edward Jenner against Smallpox, a Viral Disease. Vaccine for a viral disease, smallpox, which had devastated mankind for centuries, was developed even before the discovery of viruses. In May 1796, Edward Jenner found a young dairymaid, Sarah Nelms, who had fresh cowpox lesions on her hands and arms. On May 14, 1796, using matter from Nelms' lesions, he inoculated an 8-year-old boy, James Phipps. Subsequently, the boy developed mild fever and discomfort. 9 days after the procedure he felt cold and had lost his appetite, but on the next day he was much better. In July 1796, Jenner inoculated the boy again, this time with matter from a fresh smallpox lesion. No disease developed, and Jenner concluded that protection was complete and the first vaccine for a viral disease was developed. Self-assessment questions. 1. Which among the following statement are true? I. Viruses can be defined as large obligate, intracellular particles seen only by light microscope that have to infect and take over host cell to replicate. Answer A. False. 2. Bacteriophages infect bacteria. Answer A. True. 3. First animal virus was discovered by Twert and D. Harrell. Answer A. False. 4. Wendell Stanley for the first time crystallized virus. Answer A. True. 1.3 General account. Viruses are noncellular life forms and are obligate intracellular parasites. Viruses exist in two phases, i.e. extracellular and intracellular. In extracellular phase, the complete virus particle is called virion, which remains in an inert state, i.e., it does not carry out any metabolic activity. In intracellular phase, however, the virus replicates by using genome and metabolic machinery of the host cell. Viruses do not have organelles, cell wall, cytoplasm, nucleus, or nucleoid. Viruses have few or no enzyme of their own for metabolism. The viruses lack enzymes that synthesize proteins, nucleic acid, and those generating ATP. So a virus, in order to survive and multiply, has to use the enzymes of the host cell. The fact that viruses cannot multiply outside the host cell has considerable medical importance for the development of antiviral drugs. Antiviral drugs, as the name indicates, destroy the virus. You by now know that the virus can survive only if it is inside the cell. Let us now discuss the structural features of viruses. 1.3.1 Viral size. Viruses are so small in size that most of them cannot be seen under the light microscope, as resolution of a light microscope is limited to about 200 nm. Their size ranges from 0.02 to 0.3 μm, 20 to 300 nm, in diameter, figure 1.7. Although most viruses are smaller than bacteria, poliovirus, which is one of the smallest one, is about 28 nm in diameter. Some of the larger viruses, such as the smallpox virus, which is about 200 nm in diameter, are about the same size as very small bacteria, Mycoplasma. Figure 1.7, Comparative sizes of animal cells, bacteria, viruses, proteins. 1.3.2 Structure of viruses. The viruses are made up of only two components, A. Protein, capsid, and B. Nucleic acid, viral genome, Figure 1.8. The capsid and the viral genome are together called the nucleocapsid. The nucleic acid, DNA or RNA, is the infectious part of the virus or the part that causes diseases in the host organism. Figure 1.8, Structure of bacteriophage, tobacco-mosaic virus and HIV virus. The capsid. Capsid is a protein coat that protects the viral genome against chemical and physical damage. It also helps the virus to enter into the host cell. Capsid helps in assembly and packaging of the viral genome during viral replication, will be discussed in detail in the next unit. One of the important functions of capsid is that it induces a protective, immune, response in host. The capsid acts as antigen in the host and invokes an antibody-based immune response, which is the first layer of defense of the host from viral infection. Antibodies lock onto the outer surface proteins of a virus and prevent it from entering host cells. The capsid is constructed from a number of protein molecules, the structural subunits that are called proteomers. These protein molecules are arranged around the genome in a precise and repetitive pattern. Some viruses have single type of proteomers in their capsid. Many other viruses contain different types of structural subunits, proteomers, that assemble to form larger structural subunits of the protein coat and are called capsomers. Based on their shape and structure, nucleocapsid also gives symmetry to the virus. 1.3.3 The virus symmetry. The capsomers, as you know, are the binding blocks of capsid. These capsomers are responsible for the three different symmetry groups of viruses. The viruses fall into three symmetry groups. 1. Helical symmetry. The helical structure is cylindrical in shape, figure 1.9. The nucleic acid, DNA or RNA, and the capsomers are wound together in form of a helix. This virus structure has a capsid with a central cavity or hollow tube. The hollow tube is made by proteins arranged in a circular fashion, creating a disc-like shape. The disc shapes are attached helically, forming a tube with the nucleic acid in the middle. Viruses having helical symmetry are long rods. The length of the helical capsid is determined by the length of the viral nucleic acid, and diameter is dependent upon the size, shape, and interactions of the proteomers. Examples of a virus with a helical symmetry are the tobacco-mosaic virus, influenza virus, etc. 2. Cubica I/icosahedral symmetry. An icosahedral, icosa means 20 in Greek, is a polygon with 20 facets and 12 corners, figure 1.9. Each facet in an icosahedron is an equilateral triangle. The icosahedral forms of nucleocapsid have the shape of a regular polyhedron with triangular faces and vertices. Icosahedral capsids are constructed from knob-shaped capsomers. Each capsomer is generally made up of 5 or 6 proteomer subunits. Capsomers called pentamers, pentans, have 5 subunits, and those with 6 subunits are called hexamers, hexans. This is the most optimal way of forming a closed shell using identical protein subunits. The genetic material, DNA or RNA, is fully enclosed inside the capsid. Examples of viruses with an icosahedral structure are some plant and bacterial RNA viruses, many of which have identical proteomers in the pentamers and hexamers of their capsids. Adenoviruses are animal viruses whose capsid has 252 capsomers and its pentamers are made of different protein subunits. 3. Complex symmetry. These viruses do not show purely helical or cubical symmetry but have some extra features or are a combination of cubical and helical symmetry and may have a complex outer wall or head-tail morphology, figure 1.9. The head-tail morphology structure is unique to viruses that infect only bacteria and are known as bacteriophages. The head of the virus has an icosahedral shape with a helical-shaped tail. Figure 1.9. The 3 different symmetric groups of virus. The pox virus is one of the largest viruses in size and has a complex structure. They do not have a clear morphology, and are neither helical nor icosahedral. They lack clearly identifiable capsids but have several coats surrounding their double-stranded DNA. 1.3.4 Viral envelope. Envelope is the outer membranous layer that surrounds the nucleocapsid of certain viruses, and such viruses are called enveloped viruses. Many animal viruses and some plant and bacterial viruses are enveloped. Animal viruses like herpes virus and toga virus are enveloped icosahedral viruses. The envelope is made of carbohydrates, lipids, and proteins and are acquired from the host cell during replication. Viruses which do not have an envelope are called naked viruses or non-enveloped viruses. Since envelope is a flexible membranous structure, such viruses are usually pleomorphic, i.e., of varying shape. Their nucleocapsid can be helical or icosahedral. A layer of protein called the matrix is present between nucleocapsid and envelope. This matrix protein is responsible for holding the nucleocapsid to the envelope. The envelope of the animal virus is formed from cell and nuclear membrane of the host cell while the virus is exiting the cell via budding. Envelope proteins are coded by viral genes. On the viral envelope, special glycoproteins called spikes or peplomers are present. These spikes provide binding sites which help the virus to attach to specific receptors on the host cells. They can also be used for identification of viruses as they differ among viruses. The infectivity of enveloped viruses is mostly dependent on the viral envelope. The enveloped viruses are not able to infect host cells if the viral envelope is destroyed. Enveloped viruses are susceptible to the action of lipid solvents such as ether, chloroform, and detergents. This susceptibility of enveloped virus to lipid solvents helps us to control their spread or transmission, whereas most naked viruses are resistant to them. Thus, the 5 structural forms of viruses that occur in nature are: naked icosahedral, e.g. adenovirus, picornavirus, hepatitis A virus, naked helical, e.g. tobacco mosaic virus, bacteriophage M13, enveloped icosahedral, e.g. rubella virus, yellow fever virus, herpes virus, enveloped helical, e.g. influenza virus, and complex virus, e.g. pox virus, vaccinia virus, T4 bacteriophages. 1.3.5 The viral nucleic acid. The viral nucleic acid or genome is made of only one type of nucleic acid, either deoxyribonucleic acid, DNA, or ribonucleic acid, RNA. The DNA and RNA are made of nucleotides, which are the structural units of nucleic acids. The viral nucleic acid is usually linear or circular, and in some cases it exists as segment, influenza virus. Segmented genome occurs in fragments, as 2 or more pieces of nucleic acids and codes for various proteins. Each segment is believed to function as an individual gene. Genome size also varies to a great extent. Most DNA viruses have double-stranded DNA, dsDNA, whereas most RNA viruses have single-stranded RNA, ssRNA, as their genomic material. Most of the plant viruses are usually ssRNA viruses. Box 1.3, Structure of nucleotide. Each nucleotide has 3 parts. I. A nitrogenous base, purines and pyrimidines. The nitrogen-containing bases are cyclic compounds made up of carbon, hydrogen, oxygen, and nitrogen atoms. The purines are bases with double ring structures, namely adenine, A, and guanine, G. The pyrimidines are single ring structures, namely cytosine, C, thymine, T, and uracil, U. In the DNA, purine A, G and pyrimidine T, C are present. In the RNA, purine A, G and pyrimidine U, C are present. 2. Apentose sugar called deoxyribose or ribose sugar. 3. A phosphate group, phosphoric acid. The 2 strands of DNA or RNA are held together by weak hydrogen bonds between the nitrogenous bases of the opposite strand. Classification of viruses on basis of nucleic acid, Baltimore classification. The viruses are usually classified into 2 groups on the basis of their nucleic acid/genome, DNA or RNA, and then split into separate families based on characteristics such as strand type, double-stranded and single-stranded. 1. Viruses with DNA as their genetic material are called DNA viruses, and 2. Viruses with RNA as genetic material are called RNA viruses. The DNA and RNA viruses are further subgrouped as A. Class I double-stranded, DS, DNA viruses having double strand of DNA, e.g. adenoviruses, herpes viruses, pox viruses, etc., Figure 1.10. Figure 1.10, Class I double-stranded, DS, DNA viruses. B. Class II single-stranded, SS, DNA viruses having single strand of DNA, e.g. parvoviruses, Figure 1.11. Figure 1.11, Class II single-stranded, SS, DNA viruses. C. Class III contains viruses that have double-stranded, DS, RNA genomes, e.g. reoviruses, burnoviruses, Figure 1.12. Figure 1.12, Class III double-stranded, DS, RNA virus. D. Class IV contains viruses with single-stranded, SS, RNA genomes, positive, plus, sense. In this type of virus, the base sequence of RNA molecule is identical with that of mRNA. It means that in the host cell, viral RNA acts like mRNA and can be directly translated by the host ribosome, e.g. picornaviruses, togaviruses, etc., Figure 1.13. Figure 1.13, Class IV single-stranded, SS, RNA virus. E. Class V contains viruses that have single-stranded, SS, RNA genomes which are complementary in base sequence to the mRNA, negative, sense, RNA viruses, e.g. orthomyxoviruses, rhabdoviruses, etc., Figure 1.14. In the host cell, negative sense viral RNA acts as a template for transcription to make complementary mRNA, which is then translated by the host ribosome. Negative sense RNA viruses also carry RNA polymerase needed for transcription of their RNA. Figure 1.14, Class V single-stranded, SS, RNA virus. F. Class 6 contains viruses that have single-stranded, SS, RNA genomes that with help of reverse transcriptase enzyme convert to double-stranded, DS, DNA and then form mRNA, e.g. retroviruses, Figure 1.15. Figure 1.15, Class 6 single-stranded, SS, RNA genomes with reverse transcriptase enzyme. G. Class 7 contains viruses that have double-stranded, DS, DNA genome convert to positive, plus, sense single-stranded, SS, RNA and then with help of reverse transcriptase form double-stranded, DS, DNA and then transcribe to mRNA, e.g. hepatinoviruses, Figure 1.16. Figure 1.16, Class 7 with double-stranded, DS, DNA genome with reverse transcriptase. 1.3.6 Viral enzymes and other contents. Neuraminidase, Na, was the first viral enzyme to be discovered. You have already studied that virus cannot survive and multiply outside the host cell. For the virus to spread, it has to be released from the host cell and enter another cell. Neuraminidase is required by the virus so that it can be released from the host cell. Neuraminidase cleaves the sialic acid groups from the glycoproteins of the host cell surface. This results in the release of progeny viruses and the spread of the virus from the host cell to uninfected surrounding cells, Figure 1.17. Figure 1.17, Mechanism of action of hemagglutinin and neuraminidase. Hemagglutinin, HA, a glycoprotein has also been isolated from viruses. Hemagglutinin helps the virus to enter the host cell. HA binds with groups from the glycoproteins of the host cell surface which is responsible for binding the virus to cells and causing infection in the new cell, Figure 1.17. Another important enzyme present in viruses is the viral transcriptase, DNA-dependent RNA polymerase. Viral transcriptase is required for the synthesis of mRNA from double strand of DNA or single strand of DNA or double-stranded RNA, Figure 1.18. Figure 1.18, Mechanism of action of viral transcriptase. Some viruses also have an enzyme reverse transcriptase which synthesizes DNA from single-stranded RNA, Figure 1.19. Figure 1.19, Mechanism of action of viral reverse transcriptase. Another enzyme discovered is replicase, i.e. RNA-dependent RNA polymerase. Replicase is required for the formation of complementary RNA from single-stranded RNA, Figure 1.20. Figure 1.20, Mechanism of action of viral replicase. 1.3.7 Viral Taxonomy. The uniform classification system for classifying viruses has been developed by International Committee for Taxonomy of Viruses, ICTV. The number of viruses and their taxonomic categories are continuously expanding. The family name of viruses ends with viridae, subfamily name is virinae, and genus, and species, name is virus. For example, in the family Poxviridae, subfamily Cordypoxvirinae, the genus Orthopoxvirus contains several species of poxvirus including cowpox, vaccinia, and variola major, causative agent of smallpox. The taxonomic designation for HIV is family Retroviridae, genus Lentivirus, and species Human Immunodeficiency Virus, HIV. Viral species is a group of viruses that have same genome and same relationships with the specific organisms. Division of viruses into different taxonomic groups is based on various characteristics such as type of host organism, structure and composition of virion, nucleic acid, capsid, envelope, enzymes, strategies of replication, immunological relationship with the host, types of diseases caused and modes of transmission, and viral genome sequences. Viruses are host-specific, which means each virus is capable of infecting only certain species of host cells. Host specificity is determined by the attachment of viruses to a specific cell, which depends on the following. Asterisk specific structure of virion capsid or envelope. Asterisk presence of specific receptor sites on host cell surface, which can be recognized only by specific virions. Like, share and subscribe. What you want to watch in next video, please comment below. Keep learning, keep growing.